Antibodies bind antigens via contacts with amino acids and CDRs, but the details of binding depend upon the size and shape of the antigen. In early investigations of antigen binding to antibodies, the only available sources of large quantities of a single type of antibody molecule were tumors of antibody-secreting cells. The antigen specificities of the tumor-derived antibodies were unknown, so many compounds had to be screened to identify ligands that could be used to study antigen binding. In general, the substances found to bind to these antibodies were haptens, such as phosphorylcholine or vitamin K1. Structural analysis of complexes of antibodies with their hapten ligands provided the first direct evidence that the hypervariable regions form the antigen binding site and demonstrated the structural basis of specificity for the hapten. Subsequently, with the discovery of methods of generating monoclonal antibodies, it became possible to make large amounts of pure antibodies specific for many different antigens. This has provided a more general picture of how antibodies interact with their antigens, confirming and extending the view of antibody-antigen interactions derived from the study of haptens. The surface of the antibody molecule formed by the juxtaposition of the CDRs of the heavy and light chains creates the site to which an antigen binds. Clearly, as the amino acid sequences of the CDRs are different in different antibodies, so are the shapes of the surfaces created by these CDRs. As a general principle, antibodies bind ligands whose surfaces are complementary to that of the antibody. A small antigen, such as a hapten or a short peptide, generally binds in a pocket or groove lying between the heavy and light chain V domains. Other antigens, such as a protein molecule, can be the same size as or larger than the antibody molecule itself and cannot fit into a groove or a pocket. In these cases, the interface between the two molecules is often an extended surface that involves all of the CDRs and, in some cases, part of the framework region of the antibody. This surface need not be concave, but can be flat, undulating, or even convex. And that's antibodies bind antigens via contacts with amino acids and CDRs, but the details of binding depend upon the size and shape of the antigen. Have a great day.